students, welcome to this video. In this video, the topic is enzyme kinetics, which is very important from the exam point of view, be it any exam. And this topic, so many questions, whatever are asked, I will be covering all those questions during this lecture. So let's start. So our topic is enzyme kinetics. Kinetics means velocity or rate. Here we take the velocity or rate of enzyme catalyzed reaction, right? So in this topic, we study the effect of various variables or factors on the rate or velocity of reaction. Every time we make a graph, we'll take the factor or variable on x-axis, right? And on y-axis, velocity is taken. Velocity or rate taken on y-axis, right? Now, let's see what are the uses of enzyme kinetics. That is why we do this topic, why we study this topic. Because when we study enzyme kinetics, then we get to know the detailed mechanism of action of enzyme. In detail, we can get to know the mechanism of action of the enzyme involved, and also multiple steps which are involved in the enzymatic reaction, that also can be studied and known. Okay. Now, because of enzyme kinetics, we can do the analysis of a disease, diagnosis of a disease, treatment of a disease. How treatment? Because mostly enzymes are targets for drugs. Enzymes are targets for drugs used in treatment. Now, how, example we can take how this happens. Example, in gout, which drug we give? We give allopurinol. What is the mechanism of action of allopurinol? Allopurinol inhibits enzyme xanthine oxidase, which is the enzyme for the formation of uric acid. So the drug is inhibiting xanthine oxidase, so inhibiting the formation of uric acid in the body. So therefore enzymes are target for most of the drugs, and studying this topic will give you a very good view for the pharmacology, right? So do this topic along with the related topic in pharmacology, right? So this was that drug is affecting the enzyme, right? But also enzymes are affecting drugs in the body in vivo. How? Because many drugs are in prodrug form. Let's take an example. Example 5-fluorouracil, which is used in cancer chemotherapy, is a prodrug, right? So in body, in vivo, what will happen? It will be taken by enzymes, right? So many of the prodrugs are acted upon by enzymes so that they're converted to the active drug molecule. For example, in this case, it gets converted to the active form 2-deoxy-5-fluorouridylic acid, right? Now this compound, which is active in the body, this will be the inhibitor of enzyme thymidylate synthase, so inhibits the formation of pyrimidines, and therefore it interferes in DNA replication. Right? So here I can say that effective design and administration of pro-drug requires knowledge of enzyme kinetics, right? So that's why you need to study this topic along with link with pharmacology. Okay. So now I was telling you that enzyme kinetics, in enzyme kinetics, we study the variables. Variables are studied. So we will be taking 5 variables one by one. Which variables? First we will be taking is enzyme concentration, the effect of enzyme concentration on the velocity. Then we take substrate concentration. Very, very important. There are so many questions in this point. Then we study the effect of temperature, then pH, and then inhibitors. Okay. So let's take them one by one. First, I will be taking this enzyme concentration. So we have to see the effect of enzyme concentration on the rate or velocity of reaction. So here in this graph, we will take enzyme concentration on x-axis, velocity on y-axis, and this graph is a straight line. Basically, you have to learn the shape of all these graphs and you have to understand why this shape is there. What is the meaning of this shape? So what is the meaning? Means as we're increasing enzyme in an experiment, what is happening? We have seen the effect on velocity, that velocity is also increasing. So it is directly proportional. So velocity is directly proportional to enzyme concentration. Velocity directly proportional to enzyme concentration means if we increase enzyme concentration, rate of reaction will increase, right? So this is a linear or straight line graph, right? So let's make an equation here. V is equal to K catalysis, which is a constant, into enzyme concentration. Okay, so K catalysis is a constant value for an enzyme. Okay, so it becomes V is equal to K catalysis into enzyme concentration. Just learn this equation. I'll be using in one of the question which I'll discuss. So we are done with enzyme concentration. Coming to the second one, that is substrate concentration. So see the graph here. What I will take on x-axis? X-axis I will take substrate concentration. Y-axis I will take velocity of reaction. So this graph is like this. Like this. What is the shape of this graph? What is the shape? The shape is rectangular hyperbola graph. Rectangular hyperbola. What is the meaning of rectangular hyperbola? See here, initially when we're increasing substrate, then velocity is also increasing. Almost linear kinetics, right? But further, when we're increasing more substrate, then velocity is not increasing. See, it's a constant value here. Velocity is not increasing. So in this portion, velocity does not depend on substrate concentration, right? Why in this portion, velocity is not depending on substrate concentration? Look, this is, when I'm making this graph, this is a kind of experiment. Let's assume this we are doing in a lab. So in a lab, what we are doing? We're increasing the substrate slowly, and we are looking the velocity at each level of substrate. Okay. So here during this experiment, we will keep enzyme concentration as a constant value. We are not increasing or decreasing the enzyme concentration here. So what happens in later portion of the graph, all the enzymes active sites are already occupied with the substrate, right? So that further increase in substrate will not affect the velocity. Means at this point of the graph, all the enzymes are saturated. So we have reached enzyme saturation here, and this is maximum velocity possible. So here, this point on y-axis is the maximum velocity, that is Vmax, right? This is Vmax on y-axis. Now, let's see what is Km. If on y-axis I know the point of Vmax, I can just take half of it. Let's say this is half. So half of it means Vmax by 2, right? Now extrapolate this point on x-axis. Extrapolate this point on x-axis, then this value is Km. This value is Km. What is Km? Km is Michaelis-Menten constant. Km is Michaelis-Menten constant. This graph is known as Michaelis-Menten graph or Michaelis-Menten kinetics, right? So the graph between velocity and substrate concentration is Michaelis-Menten graph, rectangular hyperbola graph. So let's do the detail of this Km. So many questions are asked on this Km, right? So look here, can I say that Km is equal to Vmax by 2? Can I say this? Be careful. Look here, Km is on x-axis, velocity is on y-axis, so this statement is wrong. We cannot put equal sign between them. They have different units, right? What is the unit of Km? Km is on x-axis. It is representing substrate concentration. So units of Km are same like substrate concentration. It is millimoles per liter or moles per liter, right? So what exactly is Km? Km is that substrate concentration. It is that substrate concentration at which velocity of reaction is half of Vmax. Km is that substrate concentration at which velocity of reaction is half of Vmax. Learn this line nicely because usually students get confused. A question was asked, velocity at Km is option A, half of substrate concentration, option B, half of Vmax. So what will be the answer? Answer is B. Velocity is half of Vmax at Km, right? Right now you're studying the topic, so it appears very easy to you. But when in the exam you solve such questions, mostly students get confused. So please be careful. Okay, moving on further things for Km. Sometimes they ask you in the options of the question that Km, is Km an association constant? Is Km dissociation constant? Any such line written is wrong. Km is neither association nor dissociation constant. It is just Michaelis-Menten constant, right? Okay, next is Km tells the affinity. Km has a relationship with affinity between substrate and enzyme. Affinity between substrate and enzyme, and they're inversely proportional. Km is inversely proportional to this affinity between substrate and enzyme. And this Km, so if Km is large, what that means? Less affinity between substrate and enzyme. Less affinity means more of substrate concentration required to attain Vmax in that reaction. I repeat, large Km means more substrate concentration required, because now the substrate and enzyme has less affinity. So if they have less affinity, then more substrate required to bind to the enzyme, and only then Vmax can be achieved. Okay. So taking the vice versa of this, if Km is less or small, then less substrate concentration is required to achieve Vmax. That means the substrate has high affinity or more affinity with the enzyme. Okay, so Km and affinity are inversely proportional, but Km and substrate concentration are directly proportional, as Km is nothing but substrate concentration. Okay. Now, one more thing, Km, as it's a constant value, so it does not changes with change in enzyme concentration or change in substrate concentration. Km will not change with change in enzyme or substrate concentration. It's constant value, right? But in case of competitive inhibition, the affinity between enzyme and substrate decreases. Affinity decreases, so Km increases in this case. Okay, I'll take the detail of competitive inhibition in the later part of this video. Right now, just remember that Km will never change, it's a constant value. But in case of competitive inhibition, affinity decreases, so Km increases. Okay. Next, note down that Km is considered signature of enzyme. It's such a constant value that it is considered signature of enzyme. Like human beings have their signature. Signature means constant, right? So number one meaning of this signature is that, first point, that it's constant for an enzyme. Like let's say a human being, there's one person, he'll make same signature on Monday, same on Tuesday, same in January, same in February, right? So it's constant. Signature means constant. Similarly, it is constant for one enzyme, right? Second is, like, now one more meaning out of the signature word comes is that signatures are different for different individuals, right? Even twins, if twins are there, twins will also be having different signature, right? So second point which I conclude here is that Km is different for each enzyme. For every enzyme will be having a different Km value. Example, isoenzymes also, like twins have different signatures, so isoenzymes also have different Km value, like LDH1, LDH2, they both will be having different Km values. Okay, isoenzymes have different Km values. Okay, also mutation can make an enzyme with a different Km value. Even mutation can make a different mutated enzyme with a different Km value, right? So I can say that Km can be used to compare enzymes. Okay. Now let's see this rectangular hyperbola graph, and let's see which order kinetics are followed here in the graph. Look, in the initial portion, when substrate concentration is very much less than Km, then first order kinetics followed, which means that velocity is directly proportional to substrate concentration. We know that on y-axis we have velocity, on x-axis we have substrate concentration. So that is the story of initial portion of the graph. But in later portion of the graph, when substrate concentration is very much high than Km, here zero order kinetics are followed, which means that velocity is independent of substrate concentration. Okay, so learn this thing nicely. Where is first order and where is zero order kinetics and what this means. Okay, now let's do Michaelis-Menten equation. Michaelis-Menten equation. Little bit of mathematics involved here, but these days numericals have been asked in exams, so it's important to do this. Okay, so Michaelis-Menten equation, it's a rate equation for a one substrate enzyme catalyzed reaction. For a one substrate enzyme catalyzed reaction, right? And this equation tells that velocity depends on enzyme as well as substrate concentration. Okay? Earlier I told you Km will not depend on changes in enzyme or substrate concentration, right? Now I'm telling you that velocity depends both on enzyme as well as substrate concentration. So you can just directly note down the equation. V naught is equal to Vmax into substrate concentration divided by Km plus substrate concentration. This is your Michaelis-Menten equation. Now, I told you Vmax is equal to K catalysis into enzyme concentration. So we can place the formula of Vmax in this equation. So our equation becomes V naught is equal to K catalysis, which is a constant, into enzyme concentration into substrate concentration divided by Km plus substrate concentration. Okay, so you can see that our velocity depends on enzyme concentration as well as substrate concentration. Okay, so now let's take our simple Michaelis-Menten equation. V naught is equal to Vmax into substrate concentration divided by Km plus substrate concentration. And I'm going to take three situations here. First situation, substrate concentration when it is very much less than Km. When substrate concentration is very much less than Km, then Km plus substrate concentration will be equivalent to Km, right? As substrate concentration is very less, so we can just ignore the value of substrate concentration. So Km plus substrate concentration is equivalent to Km. So V naught is equal to Vmax into substrate concentration divided by Km, right? Now Vmax is constant, Km is a constant value. So we can say that velocity is directly proportional to substrate concentration. I told you this thing initially also in the rectangular hyperbola graph, that during initial portion of the graph, when substrate concentration is less than Km, then velocity is directly proportional to substrate concentration, right? Then second is when substrate concentration is equal to Km. When it's equal to Km, then V naught is equal to Vmax into substrate concentration divided by 2 times substrate concentration. as substrate concentration is equal to Km here. So you can just cut the numerator and denominator of the denominator same here, substrate concentration. So at this point, V naught is equal to Vmax by 2. So this thing also I have told you that at Km, when substrate concentration is equal to Km, then velocity is half of Vmax, right? Then third situation, when substrate concentration is very much high than Km, then you can ignore Km. Km is very less. You can ignore Km. So Km plus substrate concentration is equal to substrate concentration. Putting this in the formula, V naught is equal to Vmax into substrate concentration divided by substrate concentration, right? So we can cut substrate concentration in the numerator and denominator. So at this point, velocity is the maximum velocity, that is Vmax, and is independent of further increase in substrate concentration, right? Velocity is maximum here at this point and is independent of further increase in substrate concentration, right? So these are the three situations which we have taken and we have derived that thing. Or in short also, I have told you all these things earlier. Okay. Now there is modification of Michaelis-Menten graph known as Lineweaver-Burk plot. So let's see that. Lineweaver-Burk plot or double reciprocal curve. This is a modification. This is a modification in Michaelis-Menten graph to make it a straight line graph. Understand one concept. If some graph is a straight line graph, it's very simple and easy to do the calculations. We can make it automated also, that we can just put the values in the computer and the computer can do the calculations from a straight line graph. But Michaelis-Menten graph is not a straight line graph. It is a rectangular hyperbola graph. So it is difficult to handle the calculations here. So what scientists have done is that they have done some modification in the Michaelis-Menten graph and converted it to a straight line graph in order to make things easy. Okay, this is known as double reciprocal curve because on x-axis as well as on y-axis, reciprocal of things are taken. So let's see the graph. So y-axis, this is x-axis. I am taking the negative side of x-axis also. So this point is 0, this point is negative, right? So here it is positive direction. So the graph is a straight line graph like this. Okay. Now the slope of the graph is equal to Km by Vmax. Important. Then what is the intercept on y-axis? The intercept on y-axis is 1 by Vmax. As what we are taking on y-axis, we are taking 1 by velocity on y-axis, we are taking 1 by substrate concentration on x-axis. So slope is Km by Vmax, intercept on y-axis is 1 by Vmax, intercept on x-axis is minus. This is on the negative side of the graph. It is minus 1 by Km. This is reciprocal of substrate concentration taken, but this point represents Km. So it's minus 1 by Km, right? Now, the Michaelis-Menten equation, let's do the reciprocal of that equation. First, I'm going to write Michaelis-Menten equation. V naught is equal to Vmax into substrate concentration divided by Km plus substrate concentration. So the value is, so the equation is Vmax into substrate concentration divided by Km plus Vmax into substrate concentration divided by substrate concentration. So once I have taken Km and once I have taken substrate concentration, and the upper part is same. Okay. So here you can cancel substrate concentration. So what we get? We get V naught is equal to Vmax by Km into substrate concentration plus Vmax, right? So I'll just do reciprocal of this to give the equation of Line-Weaver-Burk plot. So 1 by V naught is equal to Km by Vmax into 1 by substrate concentration plus 1 by Vmax. So this will be the equation of Line-Weaver's-Burk plot. Very easy. Just I have deducted it from the Michaelis-Menten equation. So we have done the rectangular hyperbola graph and Line-Weaver-Burk plot. This rectangular hyperbola graph was for simple enzymes. Right? But we have other enzymes also, which are allostric or regulatory enzymes. Simple enzymes, allostric or regulatory enzymes. Most of the enzymes are simple enzymes. Few are allostric enzymes. The graph between velocity and substrate concentration in case of simple enzymes is a rectangular hyperbola graph, but the graph for allostric enzymes, the graph between velocity and substrate concentration for allostric enzymes is sigmoidal or S-shaped graph. Let's understand what are simple enzymes. See this structure. Let's say this is enzyme and this is active site of enzyme, active site where substrate will bind. So this is the substrate. Substrate can fit the active site and reaction will occur. So it's very simple here. There's no other site. There's only one site, active site. In this case, if substrate is present, reaction will occur. If substrate not present, reaction will not occur. These enzymes do not depend on any activator or inhibitor. They just depend on the presence or absence of substrate. Very simple, right? But on the other hand, allostric enzymes, they have active site, right? This is enzyme. They have active site where substrate will bind, okay? But they also have one extra site, that is allostric site or regulatory site where activator or inhibitor will bind. So here either activator will bind or inhibitor will bind. So what will happen if activator comes and binds here at the allostric site, then this binding will induce changes in the active site so that now substrate will be able to bind. Okay, so here binding depends on the presence of activator. Now, if inhibitor will bind, then what will happen? Inhibitor will bind at allostric or regulatory site. This binding will induce changes in the active site so that now substrate cannot bind. Okay, so here binding depends on the absence of inhibitor, right? Only then the binding can occur if inhibitor is not there. Okay, so these enzymes, allostric or regulatory enzymes, they are few in number and they are the regulatory enzymes of the pathway. For example, in case of glycolysis, there are 3 regulatory enzymes. I've told you that in glycolysis topic. These enzymes are PFK1, hexokinase, and pyruvate kinase. These are the 3 regulatory enzymes of glycolysis. Okay. Now let's see the sigmoidal graph of allostric enzymes on this next page. The graph is like this. Can you see the S shape? I'll make it again. Something like this. Can you see the S shape? The sigmoidal graph, right? Now, let me divide this graph into 3 portions. This much is number 1 portion. This almost vertically straight line is second portion, and this horizontally straight line is third portion. So let's understand the 3 portions of the graph. You know, this graph is between substrate concentration and velocity, right? Now, during this first portion of the graph, as substrate is increasing, velocity is also increasing directly. It is directly proportional, right? Almost linear kinetics. Then, in the second portion, now see the second portion of the graph. Okay, now see the second portion of the graph. This much substrate is increased. Only this much substrate is increased, but velocity is increased exponentially, right? This special portion of the graph is due to a property of cooperativity. Very, very important. I'll take the detail of this on next page. See the third portion of the graph. Here, we're increasing substrate, but velocity is not increasing because enzyme is saturated here. All the enzyme's active sites are occupied already by substrate. So further increase in substrate will not increase the velocity, right? Now, let's see what is this property, that is cooperativity, for allostric enzymes. Allostric enzymes, these are usually multi-subunit enzymes. They are multi-subunit enzymes. That is, they have more than 1 subunits. They have more than 1 subunits, right? Example, they have 4 subunits, right? Now, when one substrate binds to one subunit, this binding will increase the affinity. This binding will increase the affinity of other substrates to bind. Other substrates to bind. This property is known as cooperativity. These subunits cooperate with each other. They help each other in increasing the affinity for substrate binding, in increasing the speed of substrate binding, right? Like family members cooperate with each other. They are more in number, they cooperate. So here there are more subunits, they cooperate with each other in substrate binding. Okay. So because of this cooperativity property, the graph is sigmoidal. Okay. Now what will be the change in graph when activator or inhibitor comes for the allostric enzyme? So see this graph. Let's say this is the sigmoidal graph. Now, when inhibitor comes, then the graph is shifted downwards. So the graph is between substrate concentration and velocity. Now, if activator comes, then the graph is shifted upwards. Okay. So you have to learn this thing, as image-based question can be asked, and they are just shifting the graph upwards or downwards, and they can ask that the particular molecule which has affected this graph is an activator or inhibitor, right? Or you have to make this graph that which one is activator or inhibitor, right? Now, few more points about the allostric modifier. These allostric modifiers, that is activators or inhibitors, they do not bind in a concentration dependent manner. Do not bind in a concentration dependent manner. Then adding more substrate to enzyme. Adding more substrate to enzyme cannot displace the modifier. Cannot displace the modifier. What this means, let's say there are 1000 molecules of substrate and only one molecule of inhibitor. So here only one inhibitor will inhibit the enzyme and the reaction will not occur. So this modifier, that is inhibitor, does not bind in a concentration dependent manner, means even if it has less concentration, still it will bind, right? Because this inhibitor or activator, they do not bind at the active site. They instead bind at a different site, that is regulatory site. Okay. And adding more substrate to enzyme. Let's say earlier 1000 molecules of substrates are present. Now I'm making it 2000 substrate molecules. Can it displace the modifier? No, it cannot displace the modifier. The inhibitor, which is already bound, that has changed the conformation of active site so that the substrate cannot bind here. Okay. So till now we have done first variable or factor, that is enzyme concentration, linear or straight line graph. Then second variable, substrate concentration, where simple enzymes have rectangular hyperbola graph and allostric enzymes have sigmoidal graph, right? Now third and fourth I'm taking together, that is temperature and pH, as they both have bell shape of this graph. So I'm making a common graph here. Let's say this is velocity on y-axis, this is temperature on pH on x-axis, then this will be a bell-shaped graph. What is the meaning? Look, this is very low temperature or very low pH, this is very high temperature or pH, and enzymes are proteins, they get denatured in extremes of temperature or pH. You can see almost negligible velocity here during this region, right? And only in this region, which we call optimum temperature or pH, optimum temperature or optimum pH, only in this area the enzyme has velocity and the enzyme is working, right? And what is the range of optimum temperature for humans, for human enzymes? It is 45 to 55 degrees Celsius, and optimum pH range is 5 to 9 for human enzymes, right? So we are done with the 4 factors. Our fifth factor is inhibitor. Fifth factor is inhibitor. There are mainly 2 types of inhibitors: competitive inhibitor, non-competitive inhibitor. In case of competitive inhibitor, Km is increased, as competitive inhibitor binds at active site and it will decrease the affinity of substrate. It will decrease the affinity by binding at active site. So Km will be increased, but in this case velocity remains same. So Vmax is same. Non-competitive inhibitor will bind at regulatory site. In this case, Km is same and velocity Vmax will be decreased, right? So let's see the graph in case of the rectangular hyperbola graph in case of competitive and non-competitive inhibitors. So first I am making a normal graph without any inhibitor. Now in case of competitive inhibitor, Km is increased and velocity is same. This point is velocity, so let's bring our graph to this same point only. And what will happen to Km is increased. So look here, this point on y-axis is Vmax. So almost this point will be half of Vmax. This will be Vmax by 2. Now extrapolating this point, for the normal graph, if I extrapolate, this much value is Km. For the inhibitor graph, this value will be Km. So can you see Km for inhibitor is increased, right? Km for inhibitor is increased. In case of non-competitive, velocity is decreased. So somewhere here I'll bring my graph like this, right? Now this is Vmax, this is Vmax for the normal graph, right? And this point is half of Vmax for this graph, and this point is Km, right? Now this point is also, the same point is also Vmax for our inhibitor graph, right? So Vmax by 2 will come somewhere here. So if I extrapolate this point on x-axis, then the same value is Km. So you can see Km is same. Okay, Km is same. So this is the graph for non-competitive inhibitor. So here I have done the rectangular hyperbola graphs. Let's do double reciprocal graph also. In case of competitive inhibition, let's say this is normal graph. Now what effect will occur in case of competitive inhibition? Km will increase and Vmax is same. So you know this point on y-axis is velocity, Vmax. So I'm taking it same. Then Km, this point on x-axis is Km. Km is increased and this is negative side of the graph. So towards right side Km will increase. So somewhere here I'm taking the point of Km. So just join these 2 points and this will be the graph in case of competitive inhibitor, right? Now in case of non-competitive inhibitor, Vmax is decreased and Km is same. So this point is Km, I'm taking same Km, and velocity, this point is velocity on y-axis. Velocity is decreased, but this y-axis is reciprocal of velocity, so towards upper side it will be decreased, right? So just join these 2 points. This will be the graph in case of non-competitive inhibitor. Now there's one more kind of inhibitor, uncompetitive inhibitor, in which both Km and Vmax are decreased. So look at the double reciprocal graph. In this case, let's say this is normal graph without uncompetitive inhibitor. In case of uncompetitive inhibitor, Km is decreased. So this is normal Km. So towards left side it will be decreased, right? And this is normal velocity. Towards upper side it will be decreased as it is reciprocal. So join these 2 points. This will be the graph in case of uncompetitive inhibition, right? So this is it with the theory portion of the topic. Now let me tell you some questions which will help you a lot, right? So let's do the questions. So read the question. Which of the diagram illustrated below best represent the effect of ATP on hepatic PFK1, that is phosphofructokinase 1, which is rate limiting enzyme of glycolysis. Rate limiting enzyme of glycolysis. And ATP is the inhibitor of this regulatory enzyme of glycolysis. I told you, for rate limiting or regulatory enzymes, the graph shape is sigmoidal graph, and the inhibitor will bring the graph towards downside. Okay, so let's see which option from A to E. There are 5 options given. This cannot be the answer as this is not a sigmoidal graph. In case of B also, this is not a sigmoidal graph. One is a straight line. C option, they are not sigmoidal graph. E option also not sigmoidal graph. Okay, so I've excluded all the options except D. So D is the answer. And look here, this is sigmoidal graph, and blue one is the normal one. And in the presence of ATP, the graph is coming downwards. So this is due to the presence of inhibitor. Okay. Then next question. Consider a reaction that can be catalyzed by one of the two enzymes A and B. There is enzyme A, enzyme B, with the following kinetics. Enzyme A, Km is given, Vmax is given, and it is given for enzyme B also. So question number 1, at substrate concentration of 5 into 10 to the power minus 6, what will be the velocity of reaction catalyzed by enzyme A? So here substrate concentration is 5 into 10 to the power minus 6, and Km of this enzyme is also 5 into 10 to the power minus 6. So when substrate concentration is equal to Km, I told you velocity is V0 is Vmax by 2. It is half of maximum velocity. So maximum velocity is 20. 20 by 2 is 10. So answer is 10 here, right? Then question number 2, at substrate concentration of 5 into 10 to the power minus 4 moles, what will be the velocity of reaction catalyzed by enzyme B? So for enzyme B, 5 into 10 to the power minus 4 is what? Km. So here again substrate concentration is equal to Km. Velocity will be half of Vmax. Here Vmax is 30. 30 by 2, 15. 15 is the answer to this question. Then third question, at substrate concentration of 5 into 10 to the power minus 4, so this is substrate concentration. What will be the velocity of reaction catalyzed by enzyme A? Now, Km for this enzyme is 5 into 10 to the power minus 6. So which is bigger, minus 6 or minus 4? Minus 4 is bigger. So substrate concentration is more than Km. When substrate concentration is more than Km, then velocity is equal to Vmax. So for enzyme A, Vmax is 20. So answer is 20 here. So when substrate concentration is more than Km, then velocity is equal to Vmax, and this is the maximum velocity possible, and this is not dependent on this is not dependent on further increase of substrate concentration. Okay, so answer is 20 here. Let's take next question. An enzyme catalyzed reaction was carried out with initial substrate concentration 1000 times greater than Km. So initial substrate concentration is 1000 times greater than Km, right? So substrate concentration is very, very high than Km. So Km plus substrate concentration is equal to substrate concentration. As Km is very less, so we can just ignore Km value. So what will be the Michaelis-Menten equation here? V0 is equal to Vmax into substrate concentration divided by Km plus substrate concentration, that is equal to substrate concentration only. We can just cancel substrate concentration, and Vmax is equal to K catalysis into enzyme concentration. Okay, so what is my equation now? V0 is equal to K catalysis into enzyme concentration. So at this point, when substrate is very high than Km, then the short equation is V0 equal to K catalysis into enzyme concentration. As V0 is equal to Vmax, right? And I told you Vmax is equal to K catalysis into enzyme concentration. Okay, so let's read the question. After 9 minutes, 1% of substrate had been converted to product. An amount of product was 12 millimoles. if in a separate experiment, so now I'm just giving a mark here so that the next experiment is started now. In next experiment, 1/3 as much enzyme. So now we have 1/3 enzyme, and twice substrate. So substrate concentration is 2 times. Okay, twice substrate concentration is combined. How long it will take for the same amount of product to be formed? So how long means they're asking time, and the option are also in minutes. All the options in minutes. Okay, so there are two situations. So I can take one situation and two situation. So V1, V2, E1, E2. So as velocity is equal to K catalysis into enzyme concentration, K catalysis is a constant. So I can simply say V1 by V2 is equal to E1 by E2. Okay. Now E1 means initial enzyme concentration. Let's take it X. E2, in second situation, enzyme concentration is taken 1/3. They have decreased the enzyme concentration. So it is X by 3. Okay. So this is equal to 3. So what will be the velocity in second case? V2 is equal to V1 by 3. So velocity is decreased. Can you see? As enzyme is decreased, so velocity will be decreased. So in second case, velocity is equal to V1 by 3. As velocity is decreased by 3 times, so time taken will be increased. More time will be now taken for same product to be formed. So time taken, initially time taken is 9 minutes. So 9 into 3. As velocity is decreased by 3 times, time taken will be increased by 3 times. 9 into 3 is 27. That is the answer. Okay. So the question may appear difficult to you while you have not read it, but once you read it carefully and calmly, then you can easily solve it, right? So here we have just cut short the formula using this distinction that when substrate concentration is very high, then velocity is Vmax, right? Taking the next question and the last question here, very easy, but I just want to read it for you once. Allosteric modulators seldom resemble or rarely resemble the substrate or product of the enzyme. I told you allosteric modifiers, the activators or inhibitors, they bind at a site different than the active site. So obviously they will not resemble the substrate, right? So what does this observation show? Option A, modulators likely bind at a site other than active site. Correct. Option B, modulators always act as activators. No, they can act as inhibitor also. Option C, modulators bind and inhibit the enzyme. No, they can activate or inhibit the enzyme. Anything they can do, the modulator, right? Then option D, enzyme catalyze more than one reaction. No, these enzymes usually have more than one subunit, right? The reaction is only one. Okay, so answer is A only, that modulators likely bind at a site other than active site. Okay, so this is it with this video. I hope you have understood the topic well. So do revise this topic, and there's a link with pharmacology. So do read the related pharma topic also here, right? Best wishes to you.